Welcome to the SURF Introduction to Systematic Reviews of Animal Studies. This is the first module of 3, providing guidance and information on why and how to perform systematic reviews of laboratory animal research. Firstly, it's important to understand the difference between a systematic review and meta-analysis. Systematic review is a literature review to answer a defined research question. It uses pre-specified criteria to systematically locate, evaluate and synthesize evidence from scientific studies. Where appropriate, it may be followed by a meta-analysis, which is the quantitative summary of results, so the statistical pooling of results from individual studies. It may sometimes not be appropriate to conduct a meta-analysis after a systematic review, but it is not appropriate to conduct a meta-analysis without first identifying the evidence via a systematic review. Systematic reviews of clinical research, that is studies using human subjects, are well established and in the hierarchy of evidence are considered the most rigorous, particularly those that include randomized controlled trials. Systematic reviews of preclinical research, that is laboratory studies using animal models of human diseases, are relatively novel, and systematic reviews of these different types of studies serve somewhat different purposes. Although the concepts and methodological approaches of preclinical and clinical systematic reviews are somewhat similar, there are fundamental differences between the two. Clinical systematic reviews often contain fewer primary studies with potentially hundreds or even thousands of participants, whereas preclinical reviews often contain many more primary studies, but the studies themselves have only tens of animals. When we compare the purpose of clinical versus preclinical systematic reviews, there are some differences too. Clinical reviews, when they explore heterogeneity, focus sometimes on the effect of different clinical populations on the effect of an intervention. They are used to inform later phase clinical studies, and because they are considered the highest form of evidence, inform clinical practice and clinical guidelines. Preclinical evidence in exploring preclinical systematic reviews, in exploring heterogeneity, focus on the effects of different experimental designs. They may be used to inform future preclinical studies, for example, the model or the outcomes that are selected in future studies. They can inform early phase clinical trials. They have been used to explain discrepancies between preclinical and clinical trial results, and may be used to inform 3R's decisions, namely refinement and reduction of future animal studies. If we consider that we're interested in the effect of drug X on the modelling of disease Y, then we use systematic review and meta-analysis to identify all of the available evidence to support that research question. We can then assess the range and the quality of that evidence, identify potential gaps in the field, assess for publication bias, which is the phenomena that neutral or negative studies either don't get published or take longer to get published, so then they don't contribute to our synthesis of that evidence. We try to explain the discrepancies between different settings, for example, between different laboratories or between different sexes of animals, and we use systematic review to inform the design of future studies. The methods to find and select studies are intended to be transparent and reproducible, reducing bias and more likely to produce reliable and accurate conclusions. It allows us to make evidence-based inferences, and by basing decisions based on all of the available existing evidence, it prevents ongoing research waste. It's important to remember that systematic reviews are observational research. You're looking at how other people conducted and reported their experiments. So by definition, are hypothesis generating rather than hypothesis testing. Essentially, you can observe correlations between different things in the systematic review, but you cannot prove causation. So the hypotheses that led to the use or the increased use of systematic reviews in animal research were that in the life sciences, particularly in an academic setting, they are perverse incentives, things like getting research published, funded, being promoted, to produce positive results with less attention paid to the validity of these results. In the use of animal disease models, there are additional pressures to reduce the number of animals, that is due to cost, time, ethics, feasibility, that results in studies being either underpowered or of unknown power, and then these factors all combine to compromise the utility of animal models and therefore contribute to translational failure. That is translating the findings that we see in the laboratory to a clinical setting. This is an example of what we mean by translational failure, and it's taken from a systematic review looking at the totality of evidence in the modelling of stroke. In this review, over 1,000 different interventions were identified as being tested in models of experimental stroke, in vitro and in vivo models. Over 600 of these interventions were tested in animal models. 374 of these interventions were shown to be effective in these animal models. Just under 100 were taken forward and tested in humans in a clinical trial setting. However, only one intervention was shown to be effective in clinical trials. There are other existing therapeutics that are used to treat ischemic stroke, but these do not have the corresponding animal evidence to support them. So what's clear from the systematic review that was done in experimental stroke is that there are huge amounts of often confusing data, and we try to use systematic review to make sense of it. So these are data from a single systematic review, therapeutic hypothermia, to treat ischemic stroke. And we can see in this timber plot the individual experiments identified in the systematic review, all 222 experiments. There are over 3,000 animals that have been used. The vertical error bars represent the 95% confidence intervals, and the grey bar behind the graph represents the overall estimate of effect when you pull all these studies together in a meta-analysis. So what you can show is that from the published evidence, if you select extreme bits of evidence, you can either prove that therapeutic hypothermia is harmful in experimental stroke or is substantially beneficial. But by investigating the sources behind this variation, investigating the sources of heterogeneity, we might start to understand what it is about how these experiments were designed and conducted that causes these variations in the effects that we observe. And by understanding this, this may help us improve translation. There are a number of potential sources of bias in an experiment that can affect the observed results, and we use systematic review to identify the presence and impact of these potential sources of bias. Internal validity is the strength of the cause-effect relationship in an experiment. That is, are the findings that we observe due to the intervention that we've given rather than unknown systematic sources of bias? We can strengthen the internal validity of an experiment by using randomization to randomly allocate animals to experimental groups, or being blinded or unaware of experimental group allocation during the assessment of outcome. External validity is the generalizability of findings from an experiment, either to different times or different species, or even to humans in a clinical setting. Construct validity is the degree with which a model or a outcome measure encapsulate what they purport to. And publication bias is the degree with which the experiments that are conducted end up in the public domain and are not affected by significance level or positive or negative findings. So when we look at potential sources of bias in animal experiments seeking to model human diseases, we have evidence from systematic reviews across the modelling of stroke, motor neuron disease, Alzheimer's disease, Parkinson's disease, EAE, which is a model of multiple sclerosis, and glioma, and we see very few studies report measures to reduce risk of bias. Very few report randomisation, and very few report being blinded during the outcome assessment. We also can use meta-analysis to assess the impact of reporting or lack of reporting of measures to reduce the risk of bias. This is an example from a systematic review where we show the overall effect of all the studies together here in this grey bar. When we stratify according to studies that either do report or don't report randomisation, we see studies that don't report randomisation, i.e. those at higher risk of bias, reporting substantially larger treatment effects than those that do. Systematic reviews have the potential for impact to improve the reporting of animal studies. This is an example from a systematic review that we did in 2009 that we then updated in 2016. The 2009 findings show very limited reporting for measures to reduce risk of bias. In response to the 2009 systematic reviews, some authors from the majority of studies that were included in the systematic review responded, indicating that they didn't think that the fact that things weren't reported in the primary studies meant that they weren't done, but they also recognised the importance of better reporting. And this is reflected in the 2016 update, where substantially more of the studies report measures to reduce risks of bias. One of the professors whose work was highly featured in the systematic review of this research is quoted as saying that the 2009 review highlighted areas of weakness with respect to the lack of reporting on certain aspects of experimental design, and whilst it did not necessarily agree with all the recommendations, also felt that not reported did not mean not done, but they did take on board that future studies need to more fully report details, and this change is reflected in the positive outcome of the follow-up 2016 review. Systematic review has had substantial impact on clinical research. It's now routinely used by medical researchers to inform practice and policy, but also played a pivotal role in providing empirical evidence of the impact of bias in the conduct of clinical trials. These are data from a review from the early 80s that showed the difference in the number of studies that were blinded, that had blinded randomization, unblinded randomization, and no random assignment at all. We can see that as studies become less rigorous, there were a higher proportion with significant findings and with larger effect size. And this was one of the key features in driving improvements in the conduct of clinical research. When considering whether to start a systematic review, it's important to ask yourself a number of questions. For example, whether the review is necessary. You may be trying to understand the contemporary relevance of existing evidence. It may be that you're interested in the biological importance of some research. You may be trying to investigate variation in practice. Is there uncertainty and debate around some evidence, or are you trying to inform the design of a definitive study? It's also important to consider who are the stakeholders or the users of your systematic review. So who's going to use the results of your review, and are you asking questions that are relevant to those stakeholders? It's important to ascertain whether the systematic review has been done before. If it has, were the questions addressed adequately, and how recent was the systematic review performed? And it's also important to ensure that you have the necessary expertise and resources to robustly conduct the systematic review. It's important to have topic knowledge, so to understand the underlying biology around the research question, but also to have the systematic review and meta-analysis expertise to ensure that the data are identified and synthesized appropriately. So there are 8 fundamental steps in a systematic review. The first is to define the research question. The second is to conduct or write a protocol for the systematic review, where you lay out the following steps that you're going to take. You then, once you have a complete protocol, conduct the systematic search to identify the potentially relevant studies. Once you've done the search, you then need to select the relevant studies that meet your inclusion criteria, so that answer the research question that you're interested in. Once you've selected studies for inclusion, you then need to assess their study quality. You also annotate and extract information around the experimental design of the studies that you've included and the outcomes that have been reported. If appropriate, you will pull together the data in a meta-analysis and investigate sources of heterogeneity. And then finally, you will report the findings of your systematic review. We have developed some resources to support people in conducting systematic review of preclinical studies, and this is under the umbrella of SURF, our systematic review facility. SURF is a fully integrated online platform for performing systematic reviews of preclinical studies, and it's a free-to-use resource. We provide a secure screening database, data repository and analysis applications. We provide educational resources on how to conduct and report systematic review, and we provide guidance on any aspect of preclinical systematic review and the meta-analysis process. You can find details on SURF or about SURF on surf.org.uk. And if you have specific questions, then you can email us at surf.info@ed.ac.uk. In addition to SURF, there are a number of other potentially useful resources that you may wish to engage with. Circle has a website with useful tools and guidance around systematic reviews of preclinical studies. And they have a very interesting and useful infographic with the link is at the end of the slides that gives some clarity and understanding of what a systematic review is. And here are just some examples of some software that is useful in to support you conducting a systematic review. There are a number of different potential research questions that can be asked using systematic review of animal studies, and here are some examples. The first is a large-scale systematic review looking at the animal models of a specific disease, where all the evidence related to that was collected together. You can also ask a systematic review of a specific drug in a disease model. You can use systematic review to compare the effects of a drug across two different types of animal models of the same disease. You can use systematic review to assess the advantages and limitations of different models of a disease. There are, of course, as with any type of research, limitations of systematic reviews. When we assess the quality of studies included, these measures are indirect, i.e. we're looking or assessing at the reporting of measures to improve the rigour of a study. Systematic reviews are only as good as the quality of the data that goes into them. This is the concept of garbage in, garbage out. So it's important to interpret and consider the findings of a systematic review in the context of the quality of the primary evidence. There are problems or complexities around what's called collinearity in systematic reviews. So when you're looking at sources of heterogeneity, so trying to understand the aspects of experimental design that affect an outcome, if all the studies that have one variable also are the same ones that have another variable, for example, all the studies that use male animals are the ones that also blind the assessment of outcome, it limits what we can infer because we don't know whether the effects that we observe are due to the fact that the animals were male or due to the fact that the studies were blinded. Some argue that where you have too much heterogeneity, meta-analysis is not appropriate, and this is often the case in animal studies. There's lots of different experimental designs that may be answering the same research question, and essentially they're saying you are comparing apples and oranges, but it's important to consider what the purpose is of the systematic review. So in clinical reviews where you're trying to inform practice or policy, you want less heterogeneity so that you can say that the effects we see are consistent. Whereas in animal systematic reviews or preclinical systematic reviews, you're often trying to understand what the underlying effect of an experimental design choice is on outcome. So that heterogeneity actually can be useful. Common pitfalls that people make when conducting systematic reviews is that they underestimate the resources and the time required to conduct a systematic review. You require a minimum team of 3 people. This is because each step that is conducted in terms of screening for inclusion and data extraction are conducted in duplicate by 2 independent researchers, and then where there's discrepancies between these researchers, a 3rd researcher reconciles these. This is important to ensure the rigour of the findings from a systematic review. Systematic reviews can often be rapidly outdated, so it's important to consider the complexity and size of your review. Very large scale systematic reviews that take a long time to conduct, by the time you've finished, the data are already out of date. So having a very focused research question can mitigate that. If you want to get some review experience, they are often large review teams that are looking for volunteers. These are just 2 examples. There's a large COVID-19 review that is going on that's looking for reviewers, and a large review in Alzheimer's disease. These are also useful because you don't require previous experience. When you contribute to these large reviews, you are trained first before you then contribute to the live systematic review. One of the things that's important to remember is that all research is susceptible to bias. So even this type of research where you're looking at potentially other people's biases and how that affects outcomes, and you're using systematic, standardised and transparent methods, it doesn't mean that the research itself isn't susceptible to bias. And when we are looking at other people's biases, there is a risk that we don't understand and we're not self-reflective on the impact of our own biases, and we assume that discrepancies are because of other people's errors rather than that we ourselves may be biased. So if you're interested in getting involved in a large project that we're doing, a large review, here are the details. There's a guide that has been published via this link that gives details on how we are annotating the project. This is a link to the training project. We'll give you feedback. You can then join the main project and start annotating. Finally, we have a recommended reading list. We have a link to the SURF website, some publications describing methodological support for systematic reviews of animal studies. We have some example preclinical systematic reviews to give you an indication of the different types of research questions that you may answer using these methods, and a link to the Circle website that has some educational resources. Thank you very much for your time and for listening.